Hello, good afternoon, everyone. Welcome to our second session on hemodynamics. This afternoon, we're going to talk specifically about hemorrhage, how the body stops bleeding in the blood vessels to prevent blood from losing out, and um, counteracting mechanisms in which the body dissolves the clots when it is no longer needed. Um, We'll also be talking about shock and problems with um, pressure that may lead to deadly consequences. One thing that we need to understand about the body is that there are also mechanisms in place by which the body prevents blood from escaping out of the blood vessels. Blood is important because we know that it carries a lot of nutrients, it carries oxygen into the different parts of the body, and if you lose out a lot of blood, this can prove to be fatal. So the body has mechanisms in place to form plugs, to form clots, to prevent that from happening. However, we have to understand that as with practically everything, every reaction that occurs in the body, there must be a counteracting control mechanism to prevent unwanted clotting. Um, Unwanted clotting, as unwanted spontaneous clotting elsewhere in the body. Um, the formation of spontaneous clots, the formation of unnecessary clots in the body can be dangerous as it may cause blockage to the blood supply of various organs. So essentially, hemorrhage is bleeding, okay? And different types of bleeding may be manifested in different areas of the body. There may be different severities of bleeding, and bleeding um, may be a manifestation of different disease processes in the body. So, whenever bleeding occurs in mucous membranes or serosal surfaces, we classify them according to size. Small pinpoint hemorrhages are known as PTK, okay, and they actually reflect bursting of the small blood vessels or the capillaries, okay. Um, they also reflect a failure of the platelets. Okay, so these are small and pinpoint. Larger hemorrhages found in the skin and mucous membranes are known as purpura, and they are typically equal to or less than 1 centimeters. Larger hemorrhages are known as ecchymosis, and they measure more than 1 cm in diameter. And examples of these are bruises that you see whenever there is blunt trauma. Okay. Aside from the skin and the mucous membranes, hemorrhage may also occur in body cavities. Blood may collect inside body cavities, and they are known as hemothorax, hemoperitoneum, hemopericardium, or hemarthrosis, depending on where they occur. Okay. Now, the entire process by which um, blood clots are formed in order to stop bleeding and to dissolve that clot when it is no longer needed is known as hemostasis. Okay, it's a counteracting process composed of clot formation and clot dissolution. Okay, and hemostasis or the formation of blood clots tend to be divided into two. Okay, it's divided into primary and secondary hemostasis. Primary hemostasis is the initial short-acting response and is mediated by the endothelial cells of the blood vessels and platelets. Okay? The end product of hemostasis is the formation of a primary platelet plug. And while this is easily formed, it is quickly formed, it tends to be friable and it can easily be destroyed by the forces within the blood vessels, okay, by the forces of the flowing blood within the blood vessels. So as a result, the primary platelet plug has to be reinforced by multiple proteins called coagulation factors. And coagulation factors are activated sequentially with the end result being the formation of a final fibrin clot. Final fibrin clot, or the fibrin clot. The fibrin clot is the final clot which is more stable, and it is fibrin that stabilizes the primary platelet plug. And the formation of a fibrin clot is known as secondary hemostasis. Now, one of the key players of primary hemostasis are the platelets, and platelets are also known as thrombocytes. It is important to realize that platelets are not true blood cells. Okay, they are just a fragment of a larger precursor cell found in the bone marrow known as megakaryocytes. Okay, so megakaryocytes are cells in the bone marrow, and platelets are just fragments of the cytoplasm that spill out in the blood. Okay, so platelets typically last about 10 days and are biconvex discs. They are small biconvex discs. Again, they are anucleated. They do not have nuclei because they are just fragments of the megakaryocyte. Okay. Now, if you look at them under the microscope, they look like blue-violet specks, which are quite small, and they tend to clump together. Okay. Now, platelets contain granules, which contain fibrinogen, plasminogen, and thromboplastin. Okay, fibrinogen is the precursor for fibrin, which forms the final fibrin clot, and plasminogen and thromboplastin are proteins which serve to dissolve the clot. So it also has mechanisms in place to facilitate dissolution. Okay. So this is a cutout of the platelet, and it contains a cytoskeleton composed of several filaments. Okay, it contains a microtubule bundle, which serves to maintain the shape, actin and myosin, which are contractile proteins that serve to facilitate contraction of the clot. Okay, they also have a specialized endoplasmic reticulum that serves to sequester calcium. Note that calcium is a very important ion necessary for clotting. Okay? And this specialized endoplasmic reticulum is also known as the dense tubular system. Okay? Now, what happens when there is a break in the blood vessels? Okay? For us to be able to understand this mechanism, let's try to review first how a blood vessel looks like. So the blood vessel is composed of simple squamous epithelium known as endothelium. It's a single layer of squamous cells. Okay, so it's quite thin. Under that, you have the subendothelium. Okay, you have the subendothelium. Depending on the blood vessel, you will have different layers and different thickness of the layers. So the innermost layer is containing the endothelium is known as the intima. Okay, just outside the intima, you have the subendothelium, and then just outside the subendothelium, depending on whether it's a blood vessel or if it's a vein or an artery, you have a layer of smooth muscle known as media, tunica media. And then outside that, you have the tunica adventitia, which is a layer of connective tissue. Okay. Now, what is important to realize is that the endothelium and the subendothelium have contrasting properties. The endothelium secretes substances that are anticoagulant. Okay, anticoagulative. Okay, whenever the subendothelium is exposed, which is not normal, remember this: the subendothelium is and a procoagulant. So the endothelium is anticoagulant, the subendothelium is procoagulant. Normally, that should not be a problem because if the endothelium is intact, the subendothelium should not be exposed. Okay? Now, the endothelium, again, resists clotting. Okay? It resists clotting, and one of the major, most important anticoagulants secreted by the endothelium is a glycoprotein known as heparin. Okay? It's an anticoagulant. Now, by virtue that there is an anticoagulant and the endothelium has anticoagulant properties, um, blood should not normally clot. Okay, platelets should not stick to endothelium. However, when there is shear stress or any injury to the endothelium, the subendothelium will be exposed. Okay, the subendothelium will be exposed, and that facilitates platelet adhesion, platelet aggregation, and the formation of a platelet plug. Okay. Now, as mentioned, both um, the endothelium has both anti-clotting and pro-clotting mechanisms. Pro-clotting mechanisms include anti-platelet effects, substances that serve to inhibit platelet clumping, which includes prostacyclin and nitric oxide, anticoagulants like antithrombin 3 and thrombomodulin, and substances that dissolve form clots, and this include plasminogen and tissue plasminogen activators. Okay? There are also prothrombotic factors that are present specifically in the subendothelium, and the most important is von Willebrand factor. Okay, von Willebrand factor. Now, so normally, um, a platelet plug should not be formed because the endothelium secretes anticoagulant substances. Now, however, when the subendothelium is exposed, when the subendothelium is exposed, procoagulant molecules are exposed to the plasma, which contains proteins and platelets. So what's going to happen is that a molecule known as von Willebrand factor will interact with molecules in the surface of the platelets known as glycoprotein 1b. So as a result, the platelets will stick to the von Willebrand factor, and this is a process known as adhesion. Okay, so the process whereby the platelets will stick to the blood vessel wall, specifically the von Willebrand factor in the subendothelium, is known as adhesion, platelet adhesion. Now, once platelets stick to the blood vessel wall, the platelets will stick to each other as well through receptors known as glycoprotein 2b3a, glycoprotein 2b/3a. And this is a process known as platelet aggregation. Okay? Platelet adhesion and platelet aggregation leads to the formation of the primary platelet plug, and this constitutes primary hemostasis. Okay? Now, the problem with the platelet plug is that the plug tends to be friable. It can easily be broken, especially when we have fluid under pressure rushing through the vessel wall. So that platelet plug needs to be stabilized. Okay? Now, this stabilization is brought about by secondary hemostasis. What happens is that you have small proteins found in the plasma that serve to um, form the fibrin, and this is brought about by a sequential cascade of enzymes. Okay, now you have two pathways: the intrinsic pathway and the extrinsic pathway, and um, the extrinsic pathway is triggered by external substances. The intrinsic pathway is stimulated by substances that are present within the plasma. Okay, so the intrinsic pathway is initiated by factor 7 and factor 3, which serves to stimulate factor 10. Okay, the intrinsic pathway is composed of the activation of factors 12, 11, 9, and 8. And again, this activates factor 10. Okay? Now, both intrinsic and extrinsic pathway converge at factor 10. And from there, what is going to be activated is the common pathway, which is factor 10, factor 5, factor 2, and factor 1. Factor 1 is known as fibrinogen, and when it is activated, it becomes fibrin, which forms the final um, fibrin clot. Okay, so that's hemostasis. Now, there are factors which can predispose to clot formation, and these include injury to the endothelium, which makes sense because um, if the endothelium is injured, the subendothelium will be exposed. Other factors include the depletion of anticoagulants, particularly prostacyclin, when you take in medications that inhibit prostaglandin formation. Okay, so examples include NSAIDs. So this is the reason why if you take aspirin, you tend to have difficulty in forming platelet plugs, and this is the reason why aspirin is also used to prevent formation of um, clots. Okay, so if you are predisposed to heart attacks, you actually take in aspirin as part of your maintenance medications, as well as the upregulation of procoagulant substances. The presence of abnormal blood flow also creates shear stress along the wall, which creates turbulence. Okay, and once you have turbulent flow, this will damage the lining of the blood vessels. The presence of conditions that predispose you to a hypercoagulable state will also predispose you to thrombosis. And these three factors, endothelial injury, abnormal blood flow, and hypercoagulability, predisposes you to the formation of blood clots. Okay? And this is known as the Virchow's triad. Okay? Now, if a platelet plug is formed, if a platelet plug is formed, or sorry, if a clot is formed, several things can happen. The thrombus may be, or a thrombus is also known as a clot. Okay. Now, if a clot is formed, it can either dissolve through the action of enzymes. Okay, and that is good because it is no longer needed. It has stopped the bleeding, and the clot is no longer needed, and there is resumption of the blood flow. There can be organization and recanalization. So, organization is the formation of collagen around the blood clot. That's organization. Recanalization is there will be a lumen or an opening within the blood clot through which blood can flow, and this is known as recanalization. Okay? Or the thrombus can become bigger, and this is known as propagation. And what is more worrying is the clot may get pushed out by the flowing blood, and this will be carried to other parts of the body, wherein in the small vessels they may cause obstruction and ischemia, and this is known as embolization. A clot that has detached to obstruct somewhere else in the body is known as an embolus. Okay. Now, clots may form either in the venous circulation or the arterial circulation. Okay. Now, clots that form in the venous circulation include can be superficial or it can be deep. If it's superficial, they are typically painful and these are known as varicosities. Okay, so similar to what you have in your lower extremities. Um, those that are found in the deeper veins are known as deep vein thrombosis, and they are rarely painful, in contrast to um, superficial thrombosis. Okay, arterial thrombosis are much dangerous because they tend to rupture. Okay, because the blood flow or the blood pressure is greater, and once there is a rupturing of the clot, okay, this will, of course, expose the subendothelium, leading to clotting and occlusion of the vessels. Okay, now it is not just a clot that can be carried to a distant site. Generally, we call them emboli. If the embolus is from a clot, you call it a thromboembolus. Okay? But can also have fat emboli. Usually, this results from trauma to the bones, releasing the bone marrow. Remember, bone marrow is fatty, and that may lodge elsewhere in the body. Air emboli may also Air can also be cause for embolus, as well as amniotic fluid, especially when there is a rupture um, in the placenta during the birthing process. Now, as mentioned, um, a thrombus can dislodge to form blockage, and if it um, if it arises from a deep vein thrombosis, it usually blocks the pulmonary circulation. It goes to the right ventricles, and it blocks the vasculature leading to the lungs. Okay, if it lodges to the systemic circulation, then this is known as a systemic thromboembolism, and it can cause infarction, particularly in the organs where the blood vessels tend to be smaller and there are no collaterals, especially the brain, the kidney, and the spleen. Now, as a result of the blockage, infarction may occur, and how the tissue can react will depend on the nature of the blood supply. Is there only one blood supply, or are there multiple collateral blood supplies? Okay. The rate of development, or how fast the clot forms. And number three, how vulnerable the tissue is to ischemia. There are some tissues that can withstand longer periods of time without oxygen. Okay. There are also tissues that cannot last for a very long time without oxygen. This include the heart muscles, the neurons, and etc. Now, moving on to a different subsection of the topic, let's talk about shock. Shock is basically when there is um, poor circulation of blood. Okay, and poor circulation of blood or shock can be due to the heart not pumping properly, or when there is decreased blood pressure. So if the pressure is low, of course, the blood is not going to circulate. And this could lead to hypotension and um, poor tissue perfusion because blood is not circulating very well. And because there is poor blood circulation, the tissues will not get oxygenated as well. Okay? And we have general categories of shock. First is cardiogenic shock, when the heart cannot pump properly, so blood will not circulate properly. Hypovolemic shock, which is reduced blood volume. Okay, it can be due to hemorrhage. It could be due to neurogenic shock. Neurogenic shock is there is a problem with the nerves, particularly in the spinal cord, supplying the blood vessels. And because there is poor innervation of the blood vessels, they cannot control the blood pressure properly. They cannot contract or dilate properly. And anaphylactic shock, which as mentioned before, is due to the secretion of anaphylatoxins like C5a, and this is an exaggerated allergic reaction. Severe infections can also cause septic shock. So remember that cytokines, especially IL-6, are produced, which leads to vasodilation. And profound vasodilation decreases blood pressure, which leads to shock. And shock that results from infection is known as a septic shock. Okay. Apart from um, decreased blood pressure, one of the scariest things um, that can develop from shock is the presence of DIC, or disseminated intravascular coagulation. DIC results when um, DIC results when there is sepsis, and what's going to happen is, aside from the shock, the body forms small um, clots everywhere in the body. Okay, hence the name disseminated intravascular coagulation, and as a result, um, you tend to have small microclots that lodge everywhere in the body, especially in the smaller blood vessels. And by virtue that there is um, formation of clots, the coagulation factors are consumed. You run out of coagulation factors, and as a result, apart from the clotting, you also bleed out. So this is known as disseminated intravascular coagulation, and this is very fatal. And then finally, we have three stages of shock: the non-progressive, progressive, and irreversible stage. The non-progressive stage is where you have compensatory mechanisms still functional that compensate for the hypoperfusion. So the heart beats faster in the smaller blood vessels, there is vasoconstriction, and the kidneys tend to retain more fluid to try to compensate. In progressive stage, there is ischemia, starting ischemia leading to acidosis. Okay, and the compensatory mechanisms start to fall apart. And then finally, in irreversible stage, there is widespread hypoxia leading to brought about by the ischemia, and as a result, the organs start to fail. Okay. Thank you very much for listening. We'll have another session after this lecture. Thank you very much.